burning of food takes place, uh, when we say burning of food take place, so food is respiratory substrate which is going to be burned. So basically, technically, if we say it is going to be oxidized or it is going to be broken, it will be broken down or oxidized, and then production of energy will take place. So respiratory substrate get burned inside mitochondria. Formation of carbon dioxide and energy takes place, and this is called respiration. Now, what can be respiratory substrate? So respiratory substrate can be carbohydrate, fat, protein, and organic acid. So look, carbohydrate, when carbohydrate is digested, then carbohydrate will get digested. Carbohydrate will be broken down inside our digestive system into glucose, because during the process of digestion, everything or every component is broken down into the simplest form of it, or monomer. So carbohydrate, all the carbohydrate which we eat, that will get broken down to glucose. Now glucose will be taken by blood and reaches to the cells, then mitochondria. So glucose is broken down into pyruvate, and pyruvate into acetyl coenzyme, and then the Krebs cycle will start, means the respiratory part will start. Fat can be respiratory substrate. So fat will be broken down into fatty acid, then fatty acid will be converted into acetyl coenzyme, and then it will become a respiratory substrate. So finally, everything have to broken down into respiratory acetyl coenzyme. Protein, protein will break it down into amino acid, and amino acid into organic acid, and it will become a respiratory substrate. So carbohydrate can be respiratory substrate, fat can be respiratory substrate, protein can be respiratory substrate, or organic acid can be respiratory substrate. Organic acid directly enter into the Krebs cycle, right? So finally, the thing is that every substrate have to first broken down into acetyl coenzyme A or organic acid, and then it will be utilized for the production of energy. Normally, in the regular like we can say in the generic term, and the normally like we are working in a day-to-day body physiology. So normally the respiratory substrate is carbohydrate until unless there is a not any emergency situation. What does it mean? So in the normal way, respiratory substrate is carbohydrate, but if a person is going through the fasting or something else, right, going under through dieting or not getting the food, on that case, the fat will become respiratory. Suppose that a person went for the hunger strike, right? So first complete carbohydrate will be exhausted, then it will come to number of fat, then fat will be exhausted. When the complete fat will be exhausted, right, means there is no fat, then what will happen? Now this will be the turn of protein, then body will start digesting protein. When body will start using protein, this is called cytoplasmic respiration. That's what I want, right? So burning of carbohydrate and fat is called floating respiration. Please note it down. Burning of fat or carbohydrate is called floating respiration, but burning of protein is called cytoplasmic respiration, and burning of organic acid, there is nothing separate term for it. Now what happened? Formation of ATP takes place, right? So this is ATP during the burning of these substrates. Yes, Heather, any doubt?
Sir, floating respiration takes place in mitochondria, right?
All respiration takes place in mitochondria.
But
Every type
uh,
of respiration.
the protein is written as cytoplasmic respiration, so it also takes place in the mitochondria.
No. Actually, it also takes place in the mitochondria. But why we are calling it cytoplasmic respiration? Basically, what happened? The protein component are drawn from the cytoplasm, and burning of cytoplasm will take place. And when the if the cytoplasmic respiration start, it means cytoplasm itself is burning, and the organism will die. When the protein becomes substrate, then the death process of organism is started, right? This is called autophagy. Now the cell or mitochondria will start eating their own body, right? That is a condition of autophagy, and the organism will definitely die,
Okay.
right?
I get it.
Now what will happen? That mitochondria will start eating their own component, own important component, and burn it, right? So this is the process of death is start, right?
That cytoplasmic respiration is harmful for body.
Not harmful. This is the final state. It's detrimental. This process will vanish the organism. The organism have to die.
Okay.
Suppose that suppose that any person is on hunger strike, complete carbohydrate exhausted, now the complete fat exhausted. Now that person can die at any moment because autophagy have started, means body cell have started eating its own structures, their own component.
Okay, sir.
We also call this process apoptosis, या PCD, programmed cell death, or apoptosis means the death process started. It's a technical term, so out of your syllabus. So what happened? Look how the ATP generated energy. So after the burning of substrate, formation of finally ATP takes place. ATP is made up of adenosine sugar plus 3-phosphate. So that's why we call adenosine triphosphate. These bonds are made like this way. I haven't drawn a straight line because when we represent that the bond which will break down, which can be easily break down and they will release the energy. So we represent like this way. So they are breakable bond. So first ATP will break down into ADP, adenosine diphosphate. One phosphate bond will break down, one inorganic phosphate will come out and energy will be released. And this will become ADP, adenosine diphosphate. What will happen further if the energy, sufficient energy is not with the organism can break down ADP into AMP, adenosine monophosphate, right? So when this will be like ADP will be broken down into AMP, adenosine monophosphate, what will happen? One more phosphate will be broken down and the energy will be released, right? Further AMP cannot be broken down, right? Clear? So this was something basic about the respiration or respiratory part. Now let's start with the proper respiration. So type of respiration. Look. Sorry. So respiration is of first aerobic respiration and anaerobic respiration. I told you that burning of the substrate is called respiration. It may be aerobic or anaerobic. Means means oxygen is not mandatory for breaking down the substrate. In aerobic respiration, the burning of food takes place in presence of oxygen. Normally, the aerobic respiration goes on in the plant and animal cells. What happened? Glucose. So when normally we talk about the respiration, we always consider that glucose is the respiratory substrate universally. So universally, in normal situation, all the cells and all the organism uses glucose as a respiratory substrate. Glucose is oxidized, right? When the oxidation of glucose takes place, formation of carbon dioxide takes place, formation of water takes place, and 686 kilocalorie energy is utilized, right? In anaerobic respiration, what happens? In anaerobic respiration, it takes place in absence of oxygen. So normally, anaerobic respiration takes place in bacteria, fungi. It also takes place in human muscles during the hard work, running, or heavy exercise, right? When anaerobic respiration takes place in fungi and bacteria, what happens? Glucose is broken down, formation of carbon dioxide takes place, but in place of the water, formation of ethyl alcohol takes place, and only 59 kilocalorie energy is produced. So, in anaerobic respiration, less amount of energy is produced from the one glucose molecule. Anaerobic, this is a 686 kilocalorie energy, but in anaerobic, this is only 59 kilocalorie energy is produced. Now, what happened in human muscles? when when anaerobic respiration takes place in human muscle. So we are discussing that in normal situation, the respiration which will take place inside our body, that will be aerobic respiration. But what happens when we start running, when we start doing heavy exercise or some other hard work? So what happened on that time? The muscles of the body, like if you are running, your muscles of your thigh and your leg, they will require more oxygen. But heart have its limitation and lungs have its limitation. So lungs and heart are uh, unable to supply that much oxygen which is required by the muscle. So what will happen? The muscles which are not getting the oxygen in proper amount, they will start anaerobic respiration. On that condition, in our body, formation of alcohol will not take place. So what will form? So when we do the heavy exercise, for this glucose get burned and formation of lactic acid takes place. This is C3H6O3 and 36 kilocalories energy is produced. Very less amount the energy will be produced, right? This lactic acid get deposit in muscles and we feel muscular cramp. So what happens when lactic acid get deposit in our muscles? We feel muscular cramp. When we take the rest, lactic acid get broken down, and again the pain is we get the relief from the pain, right? So what is my question? Where this lactic acid is broken down? Tell me. Suppose that you have joined the wrong long run, and after the long run, you are taking the rest. Now lactic acid got deposited. Now where this lactic acid will be broken down? Yes,
So
Heather,
the
tell me.
kidney filters out the lactic acid through urine.
Lactic acid do not go on. Lactic acid is again utilized. You know, there is an organ which will take lactic acid, it will break down to pyruvate, and then again it will go for the aerobic respiration.
The liver.
Very good, excellent, very good. Liver is that organ which will take the lactic acid. Liver will break down the lactic acid into pyruvate, and again you know that the pyruvate is the substrate which is utilized in the Krebs cycle, right? Very good. Now there is a respiratory quotient. Respiratory quotient we have already discussed. I think we have already discussed. RQ is volume of CO2 evolved and volume of O2 absorbed. If RQ is high, the respiratory substrate is good. If the lower RQ, respiratory substrate is bad substrate. RQ for the glucose and carbohydrate is 1. RQ for organic acid, it is more than 1. Right? So organic acids are considered the best respiratory substrate. Remember, this can be the question. This can be the best respiratory substrate. RQ for fat is 0.7. RQ for protein is 0.9. Right? RQ you have to remember. One more important thing which I want to tell you, this question has been asked. What is the compensation point? So in a photosynthetic cell, the amount of oxygen, when amount of oxygen evolved in photosynthesis is equal to the amount of carbon dioxide produced. So what happened? The oxygen produced by photosynthesis is utilized by the respiration, and the carbon dioxide produced by respiration is utilized by photosynthesis. So intake and outtake of carbon dioxide and O2 do not take place. Means the amount of O2 which is produced will be consumed by the plant, and amount of the CO2 produced will be consumed by the plant. Right. So exchange of O2 and CO2 do not take place. This situation is called compensate point. Clear?
Yes, sir.
Is that clear?
Yes, sir.
Great. RQ, remember the RQ? Now try to understand this PPT. It will tell you what is the respiration. Only this PPT will explain everything about the respiration. Try to understand. Suppose that I have eaten chapati, or I have eaten the bread or cereals or pulses. All of them are made up of starch. They will go into digestive system. They will be broken down into simplest form, their monomer, that is glucose. Blood will absorb glucose. Blood will distribute that glucose across the body. When glucose will reach to the cells, glucose will be taken by the cell. But now the glucose cannot enter in the Krebs cycle. Krebs cycle is the cycle which will burn everything and will produce the energy. Now Krebs cycle, now glucose cannot go inside the mitochondria because the Krebs cycle goes on inside the mitochondria. So glucose is present in the protoplasm of the cell. Now the glucose will be broken down into pyruvate. The pyruvate can enter into the mitochondria, right? So glucose will be broken down into pyruvate. Glucose is 6-carbon compound. By the process of glycolysis, it will be broken down into 2 pyruvate molecule, and pyruvate is 3-carbon compound. This process of glycolysis, where the glucose is broken down, means 6-carbon compound is broken down into 2 molecule of 3-carbon compound, right? This reaction goes on in the protoplasm or cytoplasm, and formation of extra 8 ATP takes place. So glycolysis process goes on in protoplasm, right? Now what will happen? Now pyruvate enter into mitochondria because glucose cannot enter into inner membrane of, get that, glucose cannot cross the inner membrane of mitochondria. Now pyruvate can cross inner membrane of mitochondria. Pyruvate reaches to the mitochondrial matrix. This is mitochondria. This is the matrix of mitochondria. Now what will happen? Pyruvate can reach inside the matrix, but pyruvate cannot participate into the Krebs cycle. Now what will happen? The pyruvate will be broken down into a 2-carbon compound that is called acetyl coenzyme. This acetyl coenzyme may enter into Krebs cycle, and complete burning of this compound takes place due to Krebs cycle. During the Krebs cycle, formation of carbon dioxide energies take place, and this energy is utilized, right? So during the process of Krebs cycle, total 38 ATP are produced, right? During the process of respiration, 30 ATP are produced in the Krebs cycle, right? Total 30 ATP are produced in Krebs cycle and 8 from here. Then total 38 ATP are produced. Is that clear? Is it clear?
So can you explain the
Sir.
process before the Krebs cycle once?
Yes, Arpit. Be allowed.
So can you explain the process before the Krebs cycle starts?
Yes. Before the Krebs cycle, what happened? Pyruvate glucose cannot enter inside the mitochondria, is inner membrane of mitochondria. But pyruvate can enter. So pyruvate will enter into mitochondria matrix. But pyruvate is 3-carbon compound. Pyruvate cannot enter or participate into the Krebs cycle. Now what will happen? Pyruvate will convert into acetyl coenzyme, which is 2-carbon compound. Acetyl coenzyme can enter into Krebs cycle. Now acetyl coenzyme will enter into Krebs cycle. Formation of carbon dioxide and energy will take place, right? And formation of energy will take place in the form of ATP, NADPH, and FADH, right? So total 30 ATP are produced in Krebs cycle. 8 ATP are produced in where? In glycolysis. So total how many ATP are produced into burning of glucose? So from one glucose molecule, total 38 ATP energy is generated, right? Is that clear? Any doubt?
Yeah.
Okay. Now first we will discuss glycolysis, then Krebs cycle, then electron transport chain, right? Or electron transport system. Glycolysis. We divide glycolysis into 2 parts. right? Glycolysis is also known as Embden-Meyerhof pathway, EMP pathway. Both are same thing. Why? Because it was discovered by Embden, Meyerhof, and Parans, right? So Gustav Embden, Otto Meyerhof, and J. Parans discovered. So that's why it is known as Embden, Meyerhof, Parans or EMP pathway. First, what happened? First phase is called preparatory phase. In preparatory phase, the energy is spent, right? Energy is spent. So from here onward, this is the payoff phase. What will happen in the payoff phase? In payoff phase, energy is produced. So look, in preparatory phase, so we divide the glycolysis in two parts. In preparatory phase, glucose will break down into glucose 6-phosphate and 1 ATP is utilized. Glucose 6-phosphate will change into fructose 6-phosphate. Both are isomers, right? Now what will happen? Fructose 6-phosphate will break down into fructose 1,6-biphosphate. Again, 1 ATP is utilized. So these three phases are known as preparatory phase. Now from here onward, there will be payoff phase. In payoff phase, fructose 1,6-biphosphate changes into two compounds. G3P, glyceraldehyde 3-phosphate, and DHAP, dihydroxyacetone phosphate, right? DHAP will again change into glyceraldehyde 3-phosphate, G3P. Now, G3P will, what will happen? G3P will break down into 1,3-biphosphoglycerate and formation of 2-NADH will take place. Again, 1,3-biphosphoglycerate will change into 3-phosphoglycerate. Again, 2 ATP are produced. 3-phosphoglycerate will convert into 2-phosphoglycerate. 2-phosphoglycerate will convert into phosphoenol pyruvate, and phosphoenol pyruvate will convert into pyruvate. When phosphoenol pyruvate will convert into pyruvate, again 2 ATP are produced, right? So look, initially 2 ATP are utilized. You can see here the 2 ATP, 1 ATP, and 1 ATP. So I have written over minus 2 ATP, right? So minus 2 ATP. Now again, formation of 2 NADH take place. 2 NADH and formation of further here 2 ATP are produced. I have written 2 ATP. Again 2 ATP produced. I have written 2 ATP, right? So how many ATP net gain? Look, minus 2 AP consumed. 2 NADH is equal to 6 ATP. 2 ATP is equal to 2 ATP. 2 ATP is equal to 2 ATP. So in the net cell, in the balance, there will be net gain of 8 ATP in this process. Tell me, is it clear or not? And ask
So we
me
have
if
to
you
learn the
question.
enzyme.
Sorry? No,
So
no,
we will have to learn the enzymes.
don't learn the enzyme. Learn the name of only one enzyme. That have been asked twice. Succinate dehydrogenase. And that will come in the that will come in the Krebs cycle. Why? Because succinate dehydrogenase is the only enzyme which is not found in the mitochondria, but it is utilized in the Krebs cycle, and that comes from the cytoplasm. That's why that enzyme has been cast. Normally, you will go through the previous year question paper. The enzyme have not been. The only enzyme which has been asked, that the name is the succinate dehydrogenase. But why it is asked? The reason behind it, that enzyme is not found in the matrix of mitochondria. Krebs cycle goes on in the matrix of mitochondria. All the enzyme for the Krebs cycle found in the matrix of mitochondria, but succinate dehydrogenase is taken out from the outside, from the cytoplasm, right? So you have to remember only these enzyme. Need not to remember, right? Don't grab that. Is that clear? Energy balance is clear or not clear? So
Yes, sir.
in glycolysis, only 8 ATP gain. Net gain is 8 ATP, right? Anna, clear? Sania?
Can
Yes,
you
sir.
please repeat
Yes,
this
sir.
part?
Yes, yes. Who's saying?
Sir, Marita.
Yes. Pardon, please.
Sir, can you please repeat this part? The calculation of net ATP.
Fine. Yeah. I'll do that. Look, here, when glucose will convert into glucose 6-phosphate, -1 I have written over here. 1 ATP is consumed. Again, fructose 6-phosphate to fructose 1,6 biphosphate, 1 ATP is utilized. So I have written over here minus 2 ATP. Clear? This one.
Yes, sir.
Here I have written minus 2 ATP. Again, come to next. Here, when glyceraldehyde 3-phosphate changes into 1,3-biphosphoglycerate, here I have written 2 NADH are required, right? Produced. They are produced. So I have written plus 2 NADH. Again, from 1,3-biphosphoglycerate to 3-phosphoglycerate, 2 ATP are produced. I have written 2 ATP. Again, from phosphoenol pyruvate to pyruvate, again 2 ATP are produced. I have written 2 ATP, right? So minus 2 ATP minus plus 2 NADH. Now, what is the minus plus 2 NADH? So I have told you that 1 NADH is equal to 3 ATP. So look, I have copied minus 2 ATP in place of 2 NADH plus 6 ATP, right? Plus 2 ATP plus 2 ATP, right? So how many ATP?
Yes.
Total 10 is produced and 2 are consumed. So what is the net gain? Plus 8 ATP. Clear?
Yes, sir. Clear.
Okay. Remember the energy. What you have to remember? You have to remember the glycolysis cycle, not enzyme. Glycolysis cycle, where the ATP and NADH produced, and what is the energy balance sheet. The question will come from only here. Student, leave this part, but the question will come from here. You have to cram that, right? And you have to cram only those part which I am focusing. Now, Krebs cycle. I will tell you what is the energy balance sheet and all that. Look. So till now, so this was discovered by Hans Krebs. That's why we call Hans Krebs cycle, and it's also known as TCA cycle, tricarboxylic acid cycle. Right? Look, Krebs cycle will go on inside the mitochondria in the matrix part. Right? Now, what will happen? The glucose broken down into 2 molecules of pyruvate, that we have understood. Now I have told you that pyruvate can enter inside the mitochondria. Means inner membrane of mitochondria is do not allow glucose, it allow only pyruvate. Pyruvate will enter. But unfortunately, pyruvate cannot participate into Krebs cycle. For participant, pyruvate have to break down. So pyruvate will break down into 2 carbon compound, 2 molecule of 2 molecule of 2 carbon compound, that is acetyl coenzyme A. What will happen? Carbon dioxide will be released. Decarboxylation will take place. So 1 carbon molecule will be released. Formation of 2 carbon compound takes place, and the name of this carbon compound is acetyl coenzyme. And again, along with that, formation of NAD plus the NADH2 takes place. So energy is produced. Now, acetyl coenzyme A will convert it to citrate, a 6 carbon compound. Citrate will convert it to isocitrate, 6 carbon compound. Isocitrate will break down into alpha ketoglutarate. When isocitrate will break down into alpha ketoglutarate, what will happen? Again, decarboxylation will take place, means carbon dioxide will be released, and formation of NADH takes place. Mean again the energy is produced. Next. After that, alpha ketoglutarate will convert into succinyl coenzyme. Again, formation of NADH will take place, and carbon dioxide will be released. Right? Here, the enzyme which is utilized, the name of the enzyme, alpha ketoglutarate dehydrogenase. This enzyme will be produced. Again, now carbon dioxide is released, so again decarboxylation will take place. Formation of succinyl coenzyme will take place. Succinyl coenzyme A will convert into succinate, a 4 carbon. Now, what will happen when succinyl coenzyme A will convert into succinate in presence of succinyl coenzyme synthase? So, formation of GTP will take place. GTP is equal to ATP, right? Succinate will convert into fumarate, a 4 carbon compound. Again, formation of FADH2 will take place. I have told you 1 FADH2 is equal to 2 ATP. Now, succinate dehydrogenase will utilize here. Fumarate will convert into malate in presence of malate dehydrogenase, and malate will convert into oxaloacetate. Again, formation of NADH will take place, right? So this is the 4-carbon compound, and malate will convert it again oxaloacetate, and finally the oxaloacetate will again break down into acetyl coenzyme. So this is the cycle which goes on inside the matrix, and this is called Krebs cycle or tricarboxylic. Now let's calculate the energy produced over here. Try to understand. So how many ATP are produced, how many NADH are produced, and how many FADH are, right? So try to understand. In glycolysis, 2 ATP and 2 NADH2 are produced. So total 8 ATP are produced. Now here, let's count. Here, total formation of 8 NADH2 is taking place. Look how the formation of 8 NADH2 is taking place. 2 NADH2 here, 2 NADH2 here, 2 NADH2 here, and 2 NADH2 here. So total 8 NADS are produced. So look, total 8 NADH are produced. 8 3s are 24. So total 24 ATP are produced. Again, formation of 2 FADH2 will take place. 2 FADH2 is equal to 4 ATP. Formation of 2 GTP take place. So 2 GTP. So that is equal to 2 ATP. So 2 ATP, 24 ATP, 4 ATP is equal to 30 ATP. 30 ATP are produced in Krebs cycle. So try to understand. 30 ATP are produced in Krebs cycle, and 8 ATP are produced in glycolysis. So total 38 ATP are produced from burning of one glucose molecule. Clear? Go through with it and tell me. Go through with this question and tell me, is that clear or not?
So what is GTP?
GTP is equal to ATP.
They are same.
Yeah, same. Look, ATP, adenosine triphosphate, GTP, guanosine triphosphate, right? Adenine and guanine both are nitrogenous bases. Both are nitrogenous.
Yes, sir.
Sir, which are the enzymes that we have to study in this?
Here you have to remember, I was telling that this enzyme have been asked multiple times. Succinate dehydrogenase. Succinate dehydrogenase is the only enzyme. Here you can learn the enzyme because I have written the enzyme. Here isocitrate dehydrogenase, alpha ketoglutarate, succinyl co-synthase, succinate dehydrogenase, and malate dehydrogenase. So here just 5 enzyme are. You can learn the 5 enzyme at least, right? But don't forget to remember this succinate dehydrogenase. It converts succinate into fumarate, and this enzyme is not found in the mitochondria matrix. It is taken from the outside. Rest of all enzyme which are utilized in the Krebs cycle, they are found in the matrix of mitochondria, right?
Yes, sir.
Clear? Energy balance sheet clear? Look, NADH are produced. So everywhere you have to multiply by 2. Why? Because you are writing down here the one molecule, here 2 molecule. Every time 2 molecule are getting burned, right? Multiplication is 2, means 2 acetyl coenzyme will break, right? So when one acetyl coenzyme will break down, then formation of one NADH. When 2 acetyl coenzyme, then 2 NADH will. So everywhere I have make it double. Clear? So this was our Krebs cycle. So one more thing which I want to make you clear. You will see that many times in many questions and in many books, it is asked that how many ATP are produced from one glucose molecule. Sometime you will get the answer is 36, which is not correct. That is exceptional time. Normally 38 ATP are produced during the burning of glucose molecule, right? And I have proved that total 38. So I have made the balance sheet and I have told you 38 ATP. So the correct answer is 38, not the 36. Clear?
Yes, sir.
Yes, sir.
Electron transport chain. Same kind of the electron transport chain. And here I'll prove that why one NADH is equal to 3 ATP and why one FADH is equal to 2 ATP. We have to focus over here. Look, we have studied the electron transport chain or electron transport system in the photosynthesis chapter, and here also we'll see that. Look, this is the mitochondria. This is the outer membrane of mitochondria, and this is the inner membrane of. In inner membrane of mitochondria, certain cytochromes are present. This is cytochrome first, third, fourth, cytochrome third, fourth. One more thing, cytochrome first, third, and fourth, they traverse across the inner membrane. You can see they are traversing across the membrane, right? But cytochrome second do not traverse across the membrane. Actually, this is found in the inner part of the membrane, right? It do not traverse across the membrane. That thing you have to remember. First thing. Now, between outer and inner membrane, there is an intermembrane space. Look, we have done the citric acid cycle or tricarboxylic, Krebs cycle, right? In Krebs cycle, formation of GTP means ATP, formation of NADH and FADH will take place. So ATP and GTP are same. We know that. But how does one NADH is equal to 3 ATP? Look, the NADH which is formed in the Krebs cycle will break down into NAD plus and H. Now what will happen? Formation of 2 electron will take place. When these 2 electron will pass from cytochrome 1, this cytochrome will pump out 2 hydrogen ion in the intermembrane. Again, these 2 electron will reach to quinone. Quinone will pass electron to cytochrome third. When electron will pass through the cytochrome third, again 2 hydrogen ions are pumped in intermembrane state. These two electrons, when these electrons will go to cytochrome C, from cytochrome C, these electrons will go to the complex 4. When they will reach to the cytochrome complex 4, again 2 hydrogen ion pumped out, and finally the electron will be taken by the oxygen and formation of water will take place. Now you can see, due to breakage of NADH, how many hydrogen ion are pumped in the inner membrane, intermembrane space? 6 hydrogen ion, 2, 2, and 2. So due to breakage of one NADH, total 6 hydrogen ion accumulated in intermembrane space. Now, high concentration is intermembrane space, and in a matrix there is a low hydrogen ion concentration. Hydrogen ion will leak out from the intermembrane state to the matrix. When hydrogen ion will leak out, the formation of ATP will take place. So at the leakage of 2 hydrogen ion, one ATP will be produced. So at the leakage of 6 hydrogen ion, how many ATP will be produced? 3 ATP. Is that clear? That's why one NADH is equal to 3 ATP. Is it clear or not? Tell me. Arpit, is it clear?
Yes, sir.
Martina? Heather, is it clear? Abhinav? Marita, is it clear?
Yes, sir.
Anya?
Yes, sir.
Yes, sir. Could you explain the last ATP synthesis?
Yes, sure. Again, ATP synthase is an enzyme. I have told you in the last chapter, right? ATP synthase is an enzyme. This is the only enzyme through which the hydrogen ion can pass through. When the hydrogen ion, this is the only path from where the hydrogen can leak out. So when 2 hydrogen ion passes from ATP synthase enzyme, formation of 1 ATP will take place. From ATP, formation of 1 ATP will take place, right? So I told you that when electron will pass through cytochrome 1, 3rd, and 4th, every time 2 hydrogen ion are pumped in. So due to breakage of NADH, total 6 hydrogen ion are pumped in. Now, when these 6 hydrogen ion will leak, so how many ATP will be produced? So at the leakage of 2 hydrogen ion, 1 ATP is produced. So at the leakage of 6 hydrogen ion, 3 ATP will be produced. That's why 1 NADH is equal to 3. Clear?
Yes, sir.
Come to FADH. FADH will also produce. Now, when FADH2 will break down into FAD plus hydrogen ion, the electron will go to cytochrome second, but cytochrome second do not traverse the across membrane. Cytochrome second do not traverse across the membrane. So what will happen? No hydrogen ion will pump in. When 2 electron will pass through cytochrome third, then 2 hydrogen ion will pump in intermembrane space. When electron will pass to cytochrome 4, again 2 hydrogen ion will be pumped in. So when these 4 hydrogen ion will leak out from intermembrane to inner, how many ATP will be produced? So at the leakage of 2 hydrogen ion, 1 ATP is produced. So at the leakage of 4 hydrogen ion, 2 ATP will be produced. That's why 1 FADH2 is equal to 2 ATP. And it is proved. Clear?
Yes, sir.
So this is the electron transport system of mitochondria. Yesterday we did the electron transport system of the chloroplast. This is the electron transport system. Okay. So look. The same thing is here. The same thing is, I have explained the same thing with the help of this diagram, right? So the same thing which is explained over there, same thing is here, right? So don't go for that. This is very clumsy kind of. So this is the actually the part of your NCRT, and you won't get anything from this one. So better understand by these two diagrams, this diagram and with the help of this diagram, right? What is the amphibolic pathway? So first tell me what is catabolic and what is anabolic pathway? What is catabolic and what is anabolic pathway? Yes, guys.
Sir, in catabolic, complex substances are broken down into simpler substances, and in anabolic, the simpler substances are built up into complex substance.
Very good, very good, excellent. So in the nutshell, we can say that catabolic pathway is that where something is broken down. Catabolism means destruction, right? So it's a destructive. Anabolism, when something is synthesized, right? For example, the digestion of food. So larger molecule are broken down to smaller one, means something is broken down. Destruction is taking place. So that is catabolic pathway. Protein synthesis, photosynthesis, something is being formed, synthesized, bonds are made, right? So construction taking place, anabolic pathway. So where the destruction is destructive pathway, destruction is taking place, that is catabolic pathway. Where construction is taking, that is called anabolic pathway. And when the both pathway goes on in the same time, catabolic and anabolic, we called it amphibolic pathway, right? So some of the scientists says that Krebs cycle is a catabolic pathway. But most of scientists says catabolic, that Krebs cycle is not catabolic, or respiration is not a catabolic pathway. Respiration is amphibolic pathway. Why? Yes, because in the glycolysis cycle and in Krebs cycle, everything is getting broken down. Means the destruction is taking place. But acetyl coenzyme A is not only respiratory substrate for Krebs cycle, rather than it is also used as intermediary substrate or raw material for synthesis of fatty acid, steroid, terpenes, uh, uh, carotenoid, right, and aromatic substances, right? That's why this is a amphibolic path. Clear? Now look, if the digestion of carbohydrate taking place, means the starch will break down to glucose, and glucose into 1,6 phosphate, glucose 1,6 biphosphate, G3P, then pyruvic acid, acetyl CoA, Krebs cycle, energy, carbon dioxide. If fat is a respiratory substrate, then fat will break down to two things: fatty acid and glycerol. Fatty acid will directly convert into acetyl coenzyme and they will start Krebs cycle. Glycerol will convert into DHAP, dihydroxyacetylene phosphate. DHAP into G3P, then pyruvic acid, then acetyl coenzyme, and then Krebs. Protein will break down into amino acid. Amino acid will break down to pyruvic acid, and again into acetyl coenzyme and Krebs cycle. So this respiratory pathway is called amphibolic pathway, not the catabolic or anabolic. Cannot be called directly catabolic. Tell me. Clear? Is that clear?
Yes.
Yes.
Okay. Last but not least, what is the anaerobic pathway or anaerobic respiration? Look, either it's aerobic respiration or anaerobic respiration, glycolysis will go on in the same way, right, as it taken in the aerobic respiration. Now come to the anaerobic respiration. Glucose will break down to G3P, again G3P in the 3-phosphoglyceric acid, and phosphoenol pyruvate, right, in glycolysis. Now this phosphoenol pyruvate is called PEP in short. Phosphoenol pyruvate will convert to pyruvic acid. Now, when the pyruvic acid will go on for the burning in bacteria and fungi, they will form ethanol and carbon dioxide, and formation of NADH will take place, right? So formation of ethyl alcohol takes place in yeast. In yeast, pyruvic acid will convert into acetaldehyde and carbon dioxide. Acetaldehyde will break down into ethyl alcohol and NAD+. In bacteria, the pyruvic acid plus NADH is equal to lactic acid plus NAD. Formation of lactic acid will take place. In human muscles also, the lactic acid formation will take place, right? This is called anaerobic pathway. Bacteria do not require oxygen. That's why it is called anaerobic respiration. Tell me, clear? Or if you have question, please ask me. In anaerobic pathway. Done. Go through with whole chapter in next 2 minutes and let me know if you have any doubt. Alia, all clear? Samaira, Sania,
Yes, sir, clear.
Yes, sir.
good. Abhinav,
Yes, sir.
okay, great. Heather, Merita,
Yes, sir.
Yes, sir.
good. Arpit, what's about you? Clear?
Yes,
Great. So look, try to understand what I am telling you. You know, how many chapters are there in animal physiology, means human physiology? Most of the students, even who are the dropper student, they have dropped 1 year after the 12th class. They scared and they try to keep them aloof from these chapters. So there are total 5 chapters, and in the 5 chapter, almost the 4 chapter are more important. So out of these 4 chapters, they in plant physiology, they will give you 10 questions, means 40 marks. And in the human physiology, 1, 2, 3, 4, 5, 6, 7 chapters. And they will also give you the 10 questions. So the plant physiology part is more, and the chapter are shorter one. The plant physiology is more mark fetching part, and most of students leave this part. So don't leave this part. So don't leave this part, right? So go through with again. I said you go through with complete plant physiology part. Still, if you have any doubt, you are welcome with your doubt. What is the next chapter of the respiration? Plant growth and development. Basically, in the plant growth development, most of the time you will get the question from hormones, plant hormones like uh, phytohormones, and then this differentiation, dedifferentiation, redifferentiation, and SDP, LDP, and NDP example. Right, this is the only part. So it's quite easy. So this chapter is plant growth and development. Right. So first, try to understand this concept. What is the differentiation, dedifferentiation, and redifferentiation? Is there someone who want to explain me what is this differentiation, dedifferentiation, and redifferentiation? Many times this question has been asked, and it's very easy kind of a question. So I will give you example. Let me tell you, interfascicular cambium are formed by differentiation, dedifferentiation, redifferentiation, meristematic activity. That's why I have explained over it. This was the question in NEET exam. Interfascicular cambium are formed. Okay. So let me explain it. First, what is the differentiation? Formation of new cell take place from meristematic cell. So meristematic cell are those which are found in developing part, and they keep on dividing, right? So when these meristematic cell divide, then what happens? The formation of new part takes place, right? Formation of new cells takes place. So they keep on dividing and formation of new cell takes place. This process is called division. But when these new cells at maturity, when these new cells get differentiated, means they have been assigned a particular job, a particular work. This is called differentiation, right? When they have been assigned a particular job, this is called differentiation, right? So fully mature cell, they opt a particular function and lose the property of division. This is called differentiation. Now what happened? Suppose that they are a leaf cells or they are a stem cells. They are performing particular function, and that part get damaged. Wound is created over there. What will happen? Now they have lose the property of division. Now these cell will again regain the property of division, that is called dedifferentiation. What happens? You have the cells of your hand, right? There are particular cells, right? What happens? Suppose that these cells, there are particular cells. Suppose these cells have lost the capacity of division. They are not dividing right now. So they are differentiated cells. What happens? Suppose that I got injury over here. Now what will happen? The cells which have been damaged, the surrounding cell will again get the property of division. They will start dividing to fill this gap, and the process will called dedifferentiation. Dedifferentiation will take place. It again the this property it regain the property of division, start dividing, and cell become meristematic, and they will divide, right? So this is called differentiation. Now what will happen when the healing is done, right? Again, this the cells which have been which have divided, again they will get differentiated, right? When these cell will get differentiated, we will call it redifferentiation, right? Again this cell will get its function and stop dividing. So these cells are this process called redifferentiation. So that is the differentiation, dedifferentiation, and redifferentiation. Now look what happens. Normally, the cells which are there in here, the cambium part, right? So this is vascular bundle, phloem outside. This is the phloem part, right? Phloem part, and this is the xylem part, right? Between xylem and phloem, there is a vascular bundle which are already present, right? These vascular bundle between the xylem and phloem, the vascular bundle between the xylem and phloem, these vascular bundle are called intrafascicular vascular bundle. This one, they are called intrafascicular intrafascicular cambium. Intrafascicular cambium is meristematic. It have the property, the cells have meristematic. It have property of division. Now what will happen when the secondary division will take place? What will happen? These parenchymatous cells, there are some parenchymatous cells. These parenchymatous cells will regain the property of division, right? And formation of interfascicular cambium will take place, right? So formation of interfascicular cambium is dedifferentiation process, right? Dedifferentiation process. Why? Because the cells which are present over here, they are parenchymatous cells. They do not have capacity of division. These cells, when these cells regain the capacity of division and form interfascicular cambium, this property of dedifferentiation. So formation of interfascicular cambium is dedifferentiation process. Is it clear or not? Tell me. Did you got my point or not? Keep on telling me if you are getting yes. If you are not getting, say frankly no.
Yes, sir.
Very good. So this is very important part. It's simple but very important. Plasticity have been asked. What is plasticity? First, let me tell you what is heterophily. Heterophily means different type of leaf, different structure of leaf of same plant. It's called heterophyll. Look, heterophily, this A, this heterophily, this A is a larkspur and B is buttercup. Look, this is the structure of leaf, juvenile leaf. This is a structure in case of larkspur. This is a structure of juvenile leaf. This is structure of adult leaf. So at different stage of life stage, the shape of leaf is different. About buttercup, this is the terrestrial buttercup. This is the water buttercup when it is on water habitat. When buttercup is in terrestrial region, the leaf are like this way. So the habitat is different, but the same plant will have different type of the leaf, right? So heterophily is that the same species of the plant have different structure at different stage or different due to habitat. This is called heterophily. What is plasticity? When any plant changes its structure to adapt a particular environment, this property is called plasticity. Right? Clear?
Yes, sir.
Plant growth regulator. They are basically plant hormone or phytohormone. Plant hormone or phytohormone. There are 5 kind of the plant hormone. Auxin, gibberellin, cytokinin, abscisic acid, and ethylene. 5 plant hormones. They are known as phytohormones. Right? Phytohormones, just out of them, only one phytohormone, ethylene. This is the only hormone in plant and animal is found in gaseous state. It's a gas, right? The rest are just chemicals, liquid chemicals. So, what is the function of plant growth regulator? So, these phytohormones are known as plant growth regulator, PGR. Their function is growth, differentiation, and development. They differ from one organ to another organ. Means they have the different reaction at one or another organ. The similar response may also be seen in different plant growth rate, right? So the similar different plant growth hormone with different hormones, they show the similar response. The site of production and site of action may be same or different. So we will discuss one by one plant growth regulator or PGR. First, it come to the auxin. Look, the main basic function or major function of auxin that it produces phototropic response. What is phototropic response? Photo means light, tropic means in the direction of. You must have seen that sunflower always remains in the direction of sun. Suppose that sun have rise from this direction, then all the sunflower, they will have their mouth at this direction. Then they will always make their face. So in the evening, the sun will set at this direction toward the south. Then all the sunflower will have their mouth toward the south. That's why we call them sunflower, right? So they make their mouth toward their face toward the sun. How does it happen? Basically, this is the auxin which always keep the sunflower's face toward the sun. How? Suppose that this is the sun, and the face of the sunflower will always be towards sun. How it happen? This is the oxygen. Oxygen go away from the, when sunlight fall, oxygen will go away from the sunlight, right? And it will get opposite in, it get will accumulated in opposite direction, right? This oxygen will get accumulated here. What will happen when it will get accumulated here? It will enlarge the size of the cell. When cell size will be enlarged, the face of the, obviously the face of the sunflower will be at the direction of the sun, right? That's how. That's how it maintain the direction of the sunflower toward the sun. So this is called phototropic movement. So movement toward the light. So major function of oxygen is movement of plant, make the movement of plant towards the light or in the direction of light. So what are the main function? Cell elongation, increase the size of the cell. Elongation. Cell elongation is shoot. This is the hormone. This is the first discovered hormone. Now, what is very important? This question have been asked many times. You will see this question. Natural oxygen. What is the chemical composition of natural oxygen? Natural oxygen are IBA, indole butyric acid, IAA, indole acetic acid, and indole 3 acetic acid. Indole 3, right? So there are 3 natural oxygen: IBA, IAA, indole 3, right? Clog et al. Discovered the oxygen, right? And these are the natural oxygen. Synthetic oxygen. This question is also important. Both of the question, natural and synthetic, both have been asked. 2,4-D means 2,4-dichlorophenoxyacetic acid. It is synthetic oxygen, right? Means we use it artificial hormone. 2,4,5-T, 2,4,5-trichlorophenoxyacetic acid. It also have been seen. NAA, naphthalene acetic acid, right? So these all are synthetic oxygen. Clear?
Yes, sir.
Function of oxygen: stimulate respiration, cell enlargement, xylem differentiation. This question is important. I have made the star. Cell division in cambium, root initiation, right? Formation of new root. Apical dominance means this apical part will increase in the length and it will suppress the lateral growth, right? Phototropic movement, movement in the direction of light. Female flower formation in the plant. It is, if you will spray the oxygen, more female flower will be formed in the plant, right? So this was the function of oxygen. Yes. So Zipperlin. Now the next phytohormone is Zipperlin. What is the function of Zipperlin? It is weakly acidic. It increases the internodal growth, right? This part is called internode and this is node, like in bamboo. So the node where the leaves and branches come out, we call them node. And the part between two nodes is called internode, right? It was discovered by Kurosawa and Hari. Kurosawa was a Japanese farmer, basically. What is the important thing? This question have been asked multiple times. By the way, this is the older question. It was discovered by Kurosawa and Hari, right? In a fungus. The name of fungus is Gibberella fusicorae. So Gibberella fusicorae is a fungus. Please try to understand. It was discovered in so this hormone was discovered by Kurosawa, discovered in Gibberella fusicorae, which is a fungus. Even this question I have been asked, what is Gibberella fusicorae? This is a fungus. This fungus causes Bakane disease of rice. Bakane disease, it's a Japanese term. Bakane means foolish seedling disease, where the rice plant will be there, rice seedling will be there, but rice seed will not be inside there, and it will make you foolish, right? What are the function of Zibberellin? Very important. What is the baltic? So its function is growth in stem and leaves, and stem, leaves, and aerial part. It causes balting. What is a balting? Balting is the process of increasing the height of genetically dwarf plant. So normally you must have seen a cabbage always remains 6 or 9 inch large uh, rosette shaped structure. So cabbage is a plant. In the cabbage, there are leaves and they remains in the rosette, and internode are very small internode. What happened? The cabbage is maybe 6 to 12 inch, maximum 12 inch, right? But if you will express the gibberellin, this is the real picture of cabbage. You will come to know that a cabbage will become a 5. So this process is called bolting, right? Bolting is increasing the height of genetically dwarf plant. Cabbage is genetically dwarf plant. It's of 6 centimeter, 6 inch to 9 inch. If you spray the gibberellin, it will become a 5 feet or 4 feet, right? And this process will be called bolting. Gibberellin help to overcome the natural dormancy of buds, tuber, and seed. Suppose that seed, bud, and tuber, they are dormant, they are not germinating. If you spread gibberellin, what will happen? They will become active and they will germinate. So it removes the dormancy on the bud, tuber, and seed. It will causes flowering in long day plant, LDP, and it promote the male flower, right? If you spray the Berlin, what will happen? Formation of more and more male flower will take place. Is that clear? Bolting have been asked. Zibberella fusicorae have been asked. Bakane disease have been asked. Clear?
Yes.
sir.
Third plant hormone is cytokine. Zibberellin was slightly acidic, but here cytokinin is basic in nature. It was discovered by Spoog and co-worker. Spoog and co-worker discovered it. They discovered it. It promotes cell division. Right? What is the function? Function is cell division, cell elongation. If you will spread auxin and cytokinin in a combination, they will cause metamorphosis, means formation of new organ and cell differentiation. What happened? In tissue culture, what we do? We take a tissue of a plant, we multiply this tissue, and mass of a tissue means undifferentiated cells. Mass of undifferentiated, we get a mass of undifferentiated. It's a tissue, it's a mass of tissue. There is no leaf, no stem, or no leaf. If we will mix auxin and cytokinin in a combination, what will happen? Cell differentiation will take place and formation of leaves and root will take place in undifferentiated tissue or callus, right? It will cause delay or senescence, means death. It will help to overcome or seed dormancy. It is resistant to temperature. It induces parthenocarpy, means seedless fruit. If you will spray the cytokinin, formation of seedless fruit will take place, right? And it is also it also help plant to produce resistance against damage. Go through with it and ask me if you have any doubt. Cytokine plant. Clear?
Yes, sir.
Okay. Ethylene. Look, I have already told you that ethylene is the only gaseous hormone, right? It stimulates the transverse growth, right? This is the only gaseous hormone. It stimulates transverse means horizontal growth. Maximum synthesis occur during the ripening of fruit. Basically, this is the fruit ripening hormone. It is responsible for fruit ripening. Nowadays, you know how we ripen the fruit. So suppose that you can see that the banana is coming every day in market, but that much banana cannot be ripened on the tree, right? It's not possible. Practically, it's not possible. So what they do, they bring the lots of banana. They put this banana in a room, and they take the just there are some small cylinders like deodorant cylinder. They open this cylinder and throw in that room, close that room, and at the morning they will find all the banana have been ripened. So that's how they ripen the fruit, right? So ethylene is nowadays using like this way. So the ethylene is basically responsible for fruit ripening, naturally fruit ripening, right? Function ripening of fruit, and it hastens the senescence of leaves. Means if you will spray the ethylene, the leaf will fall easy, early, right? Means it speed up the ripening of leaves as well. Break the dormancy of seed and bud. Obviously, the same thing, right? Dormancy uh, breakdown, and elongation of a rise. It increases the length of rise. Responsible for root initiation, and it is responsible for to stimulate flowering in pineapple and mango. Pineapple part have been asked twice. This question have been asked twice. I've seen that. Like which phytohormone is responsible for the stimulation of flowering in the pineapple? And the answer will be ethyl. Clear? Go through with it whatever we did today, and in the next class we'll start from the uh, abscisic acid onward, right? Go through with it and ask me if you have any doubt. Done. So let's see in the next class, right?